This is lecture 13 for Biology 178, covering cardiac output and vasculature. In the previous lecture, we finished by talking about the events of the cardiac cycle and looking at diastole and systole. When we were talking about them in general, we were of course referring to the ventricles. Remember that diastole in general is referring to ventricular relaxation and filling. We can of course talk about atrial diastole, which is the atria relaxing, but diastole is generally looked at as the ventricle. Systole is going to be contraction or ejection phase. So systole is contraction, high pressure, diastole is relaxation, low pressure. And in all of these cases, we're focused on the left ventricle primarily, being that this is going to pump blood out to the body. So I know we've seen this diagram before, but I'm going to talk about it in a different way. We're going to talk about the different events of the cardiac cycle while looking at a pressure-volume relationship and to see how the pressure and volume of the left ventricle change through the different events of the cardiac cycle. You can see during late diastole, we have nice relaxation. There's going to be a volume increase in the ventricles because blood is filling them passively. From there, we will have a pressure increase as the atria contract in atrial systole, step 2, and pressure is going to drive into the ventricles with a slight increase in volume, just finishing it up. From here, we are going to have an increase in pressure, but no change in volume during number 3, the isovolumic ventricular contraction phase. This is again where we're building up pressure in the ventricles to make it strong enough to overcome the pressure inside the aorta and the pulmonary artery. From there, we reach peak pressure with ventricular ejection, and we have a drop in volume as blood flows from the ventricles out the vessels, aorta, pulmonary artery, into the body in the case of the aorta and the lungs in the case of the pulmonary artery. From there, we are going to see then a drop in that overall pressure as volume is going to start to slowly increase again during isovolumic ventricular relaxation. Now the ventricles in this stage are not filling yet, it's just the atria that are filling. So the ventricles will remain unchanged as you see that pressure kind of decline. So in the next chart, we're going to take a look at the pressure-volume relationship of these events. Now this diagram relates to the previous diagram in that it is looking at one total cardiac cycle. So all of the events that we've already discussed are here on this diagram. This diagram is simply looking at the relationship between pressure, which is going to be on your y-axis, and volume, which is on your x-axis. If you're following along with your lecture notes, this is actually what I would like you to draw in that space. First, let's start with the purple square A. A to A, so that point A to the second point A, represents late diastole. This, of course, is going to be the passive filling of the left ventricle, being that the atrioventricular valves are open, blood is flowing into the atria and just passively flowing into the ventricles. From there, we move from A to B. A to B shows us we have an increase in volume and an increase in pressure, whereas from A to A, all we saw was an increase in volume with no increase in pressure. So A to B represents the actual filling of the ventricles via active contraction of the atria, or the start of atrial systole. Then we go from B to C. B to C actually shows us a constant volume, but an increase in pressure. So with a constant volume that is unchanged, but an increase in pressure, this can only mean this is the isovolumic contraction phase, where there is no movement occurring, but pressure is gradually increasing in the left ventricle. Once we get to C, we are now at the end diastolic volume. We're finishing the diastole stage and beginning to start the systolic stage. So C, end diastolic volume. This is about 135 milliliters in a 155-pound man. We'll then go from C to D. This is ventricular contraction or the ejection phase. From this point, C to D, we notice there is a decrease in volume, but a sharp increase in pressure, which makes sense. The ventricles are contracting and emptying the heart out. When we get to D, this represents the end systolic volume. This is approximately 65 milliliters in that same 155-pound man. From D to A, we then go through ventricular relaxation, where we are decreasing the pressure with volume being unchanged. So the valves are closing, both the semilunar and the atrioventricular valves. We've heard the second sound, the dup sound of the semilunar valves closing, and the heart is relaxing as the atria begin to fill with blood, but the ventricles are not filling with blood as it moves from D to A. Like I said before, looking at C and D, we can see C is end diastolic volume and D was end systolic volume. When we subtract the end systolic volume, the end diastolic volume minus the end systolic volume, we get what is called stroke volume. Stroke volume is the amount of blood that is pumped by one ventricle during a contraction. So the stroke volume is going to average about 70 milliliters per beat. That's going to be its measurement. And we know from previous lecture that our heart rate is actually going to be, on average resting heart rate, 70 beats per minute. 70 times 70, that's right, it's 4,900 milliliters per minute, because that's what we're looking at, and that's called cardiac output. This is the volume of blood pumped by one ventricle in a given period of time. In this case, generally a minute. And this averages about 5 liters per minute in a normal individual. You will need to calculate cardiac output for the exam as well as stroke volume. So make sure you know these formulas. Stroke volume is end diastolic volume minus end systolic volume. And cardiac output is heart rate multiplied by stroke volume. So we've looked at cardiac output, which is heart rate times stroke volume. And we said that heart rate is going to be 70 and stroke volume is going to be 70. So 70 beats per minute and then 70 milliliters per beat. But this, of course, is at rest. Well, throughout your day, there are times when your body puts greater demands on your heart than at other times. So, of course, it's going to naturally increase in heart rate or cardiac output, and it also might decrease cardiac output as well. An example would be when you're walking up the stairs or running to catch a train versus sitting in class. Now, the question becomes, how does your body regulate this different type of cardiac output? Well, it does this via the autonomic nervous system, as we've discussed in Section 1. The autonomic nervous system is broken down into the sympathetic and parasympathetic divisions. We're going to talk about how both of these divisions interact with the heart and regulate cardiac output. Sympathetic neurons use norepinephrine on beta-1 receptors, and parasympathetic neurons are going to use acetylcholine on the muscarinic receptors. I'm not going to require that you memorize these different receptors or neurotransmitters, but I just want you to understand the different variables that play into it. We're going to start with sympathetic nervous system. The sympathetic nervous system is going to increase cardiac output. It does this first and foremost by increasing heart rate. This wave should look a little familiar to you. It is actually going to be the electrical activity of the pacemaker cell, the SA node. The key thing to note here is the gray line first is going to represent that normal 70 beats per minute depolarization of the SA node. The red line is going to represent what's going to happen with sympathetic stimulation. Sympathetic stimulation and epinephrine are going to depolarize the autorhythmic cell and speed up the pacemaker action potential. It does this by actually increasing the leakage of sodium and potassium in the cells, thus allowing it to reach threshold faster and not to return down as low. So it's actually bringing the resting membrane potential of the cell up slightly. Epinephrine and norepinephrine from the sympathetic nervous system have more than just an effect on heart rate. They also increase the force and velocity of contraction. It goes through a general process in the heart after norepinephrine and epinephrine are released, binding to receptors and activating different protein signals in the body. These protein signals are going to cause two things to happen. I don't need you to memorize what's on this picture. It's simply visualizing these two events. It increases the speed of the sarcoplasmic reticulum calcium ATPase. This, of course, is going to be the pump that's going to pump calcium back into the sarcoplasmic reticulum, if we remember from way back when we talked about the muscular system. This makes the cells relax faster so they can contract again. It's also going to superload and open up the ryanodine receptor for a longer period of time. This is going to release more calcium. So epinephrine and norepinephrine are going to cause more calcium to be released from the sarcoplasmic reticulum and cause it to be re-uploaded in the sarcoplasmic reticulum faster. These two variables are going to help to increase the force and velocity of contraction by affecting the movement of calcium. And that's what this diagram is looking at, is norepinephrine and epinephrine binding to those receptors, activating that messenger system in the cell, thereby causing the sarcoplasmic reticulum to release more calcium and to uptake it faster. It's giving those secondary signals to increase that rate. From sympathetic, we'll now move to the parasympathetic neurons. Parasympathetic in general, if you remember, is rest and repose, or rest and relax. So that means the parasympathetic nervous system is going to slow down or decrease heart rate. As you can see in this diagram, it's going to have more space between the beats. Like I said, parasympathetic nervous system stimulation is going to slow down the heart rate. Here we can see the gray line, which represents normal heartbeat or SA node depolarization, and we can see the blue line, which is parasympathetic stimulation. The activation of the parasympathetic nervous system and the release of acetylcholine onto the receptors of the heart causes the pacemaker cells to hyperpolarize, and by hyperpolarizing, it takes longer for those leak channels to actually cause it to reach threshold. This will cause a slowdown in heart rate. This is actually the only effect that the parasympathetic nervous system has directly on cardiac output. It can't affect contractility the way that the sympathetic nervous system does. But suffice it to say, when parasympathetic nervous system is activated, it's going to bring down the actual heart rate, the cardiac output, through decreased stimulation of the SA node. And additionally, it's going to not allow the contractility of the heart to increase. So it's going to just be at normal contractility function. This is a diagram looking at everything that we've just talked about in the previous slides. It's simply looking at the control of the cardiovascular system, which is controlled by the medulla oblongata. Remember, that's going to be at the base of the brainstem, serving a lot of our very important autonomic functions like cardiovascular heart control and blood pressure control. And it's divided into parasympathetic and sympathetic division. The parasympathetic neurons that we just talked about are going to involve muscarinic receptors of the autorhythmic cells, and they cause the stimulation of those causes a potassium influx and a decrease in calcium. This is going to hyperpolarize the cell and decrease the rate of depolarization, thus decreasing heart rate. So that's what we just talked about in the overall effects that we'll have. What we talked about a little bit before that was sympathetic neurons, using, of course, norepinephrine and epinephrine paired, and it's going to be activating the beta-1 receptors of the autorhythmic cells. This causes an increase in sodium and calcium, thus increasing rate of depolarization and increasing heart rate. This is just looking at the effect on heart rate overall and not discussing the contractility function that we've also mentioned that sympathetic neurons have an effect on. The heart can obviously regulate its contractility through sympathetic nervous system intervention, but it can also regulate heart contractility through an intrinsic regulation mechanism. This intrinsic regulation mechanism is called the Frank-Starling mechanism. The Frank-Starling law states that stroke volume increases as end diastolic volume increases. If you remember, end diastolic volume is going to be the amount of blood that's in the ventricle before it contracts. That makes sense. I'm going to pump more blood per beat if I have more blood in the ventricle. How do we actually increase end diastolic volume, or the amount of blood in the ventricle at the end of relaxation? Well, we do that through venous return. Venous return is about bringing blood back to the heart, eventually through the vena cava and the right atrium. Venous return is affected by three main variables. The first is the skeletal muscle pump. The skeletal muscle pump is effectively your muscles contracting and relaxing. And every time that they contract, they are not just moving, and they're not just moving your body. That by them contracting, they actually squeeze your veins, which pumps blood back towards the heart because it's a one-way system. So it's helping to increase blood flow. This is why that if you ever feel like your legs are, like pooled with blood from sitting for a very long time in the car, what do? You get out, you walk around, and suddenly you can feel that the blood is starting to move from your legs and back towards the heart. So that's a skeletal muscle pump. The next is the respiratory pump, and this is exactly what it sounds like. It involves your lungs. The faster you breathe, the faster air is going to move into your lungs and transfer with the blood. So if you're increasing respiratory rate, then you're increasing the speed within which blood moves through your respiratory system, the pulmonary circulation circuit. And if you increase that, it's going to move the blood faster, thus making venous return greater. And finally, we have a sympathetic innervation of veins. This is going to actually be the constriction of large veins, blood reservoirs, to allow that pressure to control gradually and keep the pressure gradient going of the veins from that high pressure to low pressure. So it's going to constrict some, increasing the pressure in some areas and then relaxing it as it moves through. So it's going to help to increase overall blood flow. This increased venous return stretches the chambers of the heart, which will have the following effects. It will stretch the contractile myocytes to improve overlap between actin and myosin so that tension developed improves. Stretching of the myocardial cells, both the contractile and the pacemaker, also increases the movement of calcium and sodium into the cell. This will cause a more rapid depolarization, so it's going to increase heart rate, and a greater intracellular calcium release for an increased contraction strength. So that means the stretching of the heart is going, every time you stretch the heart, it will contract stronger and start to beat faster. So the Frank-Starling law, like it states here, stroke volume increases as end diastolic volume increases. This is because the more that you stretch the heart, the harder it will contract and the faster it will contract. So now let's circle back a little bit and look at everything that we've covered and all the variables that affect cardiac output. Remember, cardiac output is calculated as stroke volume multiplied by heart rate. And remember that stroke volume is end diastolic volume minus end systolic volume. So cardiac output is a function of heart rate and stroke volume. Heart rate is determined by the rate of depolarization in the autorhythmic cells. If we decrease the heart rate, by decreasing heart rate, we're decreasing the rate of depolarization of these cells. We do that through parasympathetic innervation. If we're increasing the rate of depolarization, this is done through sympathetic innervation and epinephrine. Let's go over to stroke volume now. Stroke volume is determined by the force of contraction in the myocardium, and we're talking specifically about the ventricular myocardium. This is influenced by the contractility, so the ability of the actual actin and myosin to be able to bind and slide across each other, and then the end diastolic volume. Remember that contractility can be increased through sympathetic innervation, which we talked about is controlled by calcium release. Sympathetic innervation also is going to have an effect on venous constriction, which increases rate of venous return. This, of course, affects end diastolic volume. Venous return is aided by the skeletal muscle pump, respiratory pump, and then the venous constriction that we already talked about. So these overall factors are all going to contribute to the cardiac output. So we finally finish with the heart. We're going to talk about it a little bit more in the next lecture with respect to blood pressure. But when we're talking about blood pressure, we need to talk about what blood moves through. Now, the chambers of the heart don't work alone. They need blood vessels. To understand how blood vessels contribute to the movement of blood through the cardiovascular system, we need to look at the different types of vasculature that we have. So on the right column, you'll see a nice red color. This is representative typically of arteries. In many models or diagrams, arteries are usually classified as red. This is because typically, but not always, arteries are going to carry oxygen-rich blood, denoted by the red color. However, in pulmonary circulation, remember that arteries carry deoxygenated blood. The definition of an artery is that it always carries blood away from the heart. That's the key thing to remember about arteries. It's also going to be, arteries are going to be a higher pressure system. And you can see that as we work our way down with the arteries, they have a very thick wall as far as their overall vessel. Let's contrast that with the veins. Veins always carry blood towards the heart. And typically they're seen as blue because typically in systemic circulation they carry deoxygenated blood. The case where this is not correct is going to be in pulmonary circulation. Always remember that systemic and pulmonary, it's going to be flipped as far as which vessel carries oxygenated blood. So the veins are actually going to carry deoxygenated blood throughout the body, but oxygenated blood from the lungs back to the heart. The key definition of a vein is veins always carry blood back towards the heart. We can see that veins have a very wide lumen or opening and very thin walls by comparison to arteries. The final vessel that we can see there are capillaries. Capillaries are going to be where nutrient and waste exchange occurs, so they're very, very thin. We can grow new capillaries rapidly, and they exist throughout the body, connecting to every tissue. We're going to first look at the histological organization of blood vessels and the different layers. They come in 3 layers. First is the tunica intima. This is the inner layer. It has an endothelial lining and an elastic connective tissue. In arteries, it has a slightly thicker amount of elastic fibers, giving it a wavy appearance. The middle layer is the tunica media. This is smooth muscle and connective tissue. Arteries may include an additional band of elastic fibers or an external elastic membrane in this layer as well. The smooth muscle contraction causes vasoconstriction, and the relaxation of this smooth muscle causes vasodilation. Typically, arteries have a larger overall amount or a thicker amount of smooth muscle in this layer and the thickest of the tunica medias. The final layer is tunica adventitia. This is the outer layer. It is composed of dense, irregular connective tissue sheath containing collagen, elastin fibers, nerve endings, and different macrophages. It plays an important regulatory role over the function of the muscular media and controls the leakiness of the endothelium. How do we distinguish arteries from veins? Most arteries and veins will run parallel to each other in the body. That makes it really easy to learn when we're learning location. So the brachial artery is right next to the brachial vein, the axillary artery next to the axillary vein. Even the aorta and the vena cava run next to each other. Arteries will always carry blood away from the heart, and veins carry blood towards the heart. That's a key distinguishing feature. The walls of the arteries are much thicker than that of veins. Arteries are going to maintain their shape, whereas veins will collapse when cut. If we were able to actually feel the difference between arteries and veins, arteries are going to feel almost slightly rubbery. You can feel how thick they are. Veins feel almost papery because of how thin their walls are. The endothelial lining of an artery also has pleated folds. Here we can see an artery compared to a vein. Note some of the key distinguishing features we talked about. First, the artery, much thicker, muscular middle layer there. And it also is going to have those pleated folds on the inside, whereas the vein does not. We can see that in the actual slide in the center, the artery has maintained a lot of its circular shape, whereas the vein is kind of collapsed over and just goes with the flow, so to speak, as far as the other tissues around it. Here we can see the structure of an artery wall. I want you to just take note of how thick the smooth muscle is and that pleated endothelial layer because of the internal elastic membrane. So we've covered arteries and veins. I now want to talk about capillaries. We'll talk about capillaries in much greater depth, including how their exchange works and the different types of capillaries in lecture 15, but I want to talk about some basics right now. First off, capillaries are very, very thin walled. They're one cell wall thick, and they are this thin in order to permit exchange of gases between the blood and the interstitial fluid, because this is where exchange happens. The diameter of a single capillary is about that as a red blood cell. So they are not only do they only have like this thin wall, but their overall diameter only allows one cell through at a time. So again, very, very small. There's specialized capillaries, also called fenestrated capillaries. These contain larger pores or openings in the capillary walls. So this is going to be a typical capillary that we would see. It's going to have that endothelial cell that's going to be the lining, and then a basal lamina that surrounds that and fuses that to the rest of the tissue. If we zoom in, we can actually see that there's going to be exchange that occurs through the endothelial wall, but if it's a fenestrated capillary, it's also going to have these openings or pores that would allow for larger overall exchange. This is going to be seen in different areas like the liver, things like that, where more exchange is necessary. Now, capillary beds are going to be an interconnected network of capillaries. We continuously grow new capillaries as our tissues grow and develop. When we say that when we work out, for instance, you're going to increase your overall vasculature because your body is using that tissue more, so it needs to fuel it more, so it's going to grow more capillary beds, these networks of capillaries that connect out to the tissue. Capillary beds consist of vessels connecting arterioles, small arteries, and with venules, small veins. There are precapillary sphincters involved in regulating blood flow through these capillaries. So this gives you a sample of what a capillary bed would look like. We can see the arteriole on one side and the venule on the other. On the arterial end, we can notice we have precapillary sphincters. These are going to be all around surrounding the different parts of the capillary bed. Here we have a nice open capillary bed and exchange happening. This is going to be that nice open capillary bed. We can see blood flowing through, all free, when precapillary sphincters are relaxed and blood flows just through the capillary bed. However, if the precapillary sphincters constrict, we restrict blood flow to this capillary bed, and it still runs through the area, but it's not going to be going out as much. We're able to actually redirect blood flow. This is an example of how autonomic nervous system regulation would help to control where blood flow goes. So when you have sympathetic nervous system stimulation, your body actually is going to redirect blood flow from a lot of your digestive tract or digestive functions and put it towards your muscles. Like a tiger comes in, oh my gosh, it's a tiger. And all of a sudden you start to feel more blood rush to your legs. Your body's getting ready. I need to fight or flight. It's using the precapillary sphincters, shutting down digestive function to a degree, not all the way. There's still some blood flow there, but it's restricting some blood flow and then opening up the precapillary sphincters in your muscles. Flip that over to you're having a nice day of relaxing on the couch. Your body is not going to be sending as much blood to the muscles. It's going to be focusing on your digestive tract or even neurological function. So it's going to open up the blood vessels in your digestive tract and then restrict the blood vessels in some of your legs down to base levels just to maintain the tissue. Now, blood in the veins returning to the heart from the lower extremities has to go against gravity. To assist in this process, many veins have valves. So these are venous valves, and they're similar in overall structure to some of the heart valves, just as far as being kind of like flaps of connective tissue. These valves compartmentalize the blood in the veins, thus acting as a one-way system. In the venous valve, if the venous valves do not close properly, varicose veins may occur. So this is a sample of two things. First, how the valves work. We can see here a valve opens above the contracting muscle to allow blood to move out and closes below the contracting muscle to force blood in place and make sure that it doesn't backflow. So these are how valves are going to compartmentalize blood and pump it up. The other thing that this diagram shows is the skeletal muscle pump. So as the muscle contracts, it's going to essentially milk blood back towards the heart and use the valves to prevent the backflow. In the next lecture, we're going to talk a little bit more about blood vessels and the regulation of blood pressure.